Hey science buffs, it's Kaustav from Biogenius ATS, and today we are delving into the dynamic world of cellular chemistry in our video Fate of Pyruvate. If you are new here, smash the subscribe button and hit the bell icon. It's your VIP pass to our science adventures. To our regular people, welcome back. Your hunger for knowledge keeps this channel buzzing. And before going any further, please check my last video on glycolysis for the better understanding of this video. So without wasting any time, let's roll. So what is pyruvate? Pyruvate is a key molecule in biochemistry and cellular metabolism. It is a 3-carbon compound that plays a central role in various metabolic pathways within the cells. Pyruvate is produced during the breakdown of glucose through a process called glycolysis, which occurs in the cytoplasm of cells. Pyruvate is a versatile compound because depending upon the cell type and the availability of oxygen, it shows different reactions and outcome. So let's talk about the fate of pyruvate. So in cells, pyruvate mainly has two peculiar fates. One, pyruvate to lactate, and two, pyruvate to acetyl-CoA. In this video, we will be discussing about the first fate of pyruvate, that is lactic acid formation, and how this lactate is reused further. So pyruvate, as I mentioned above, has two fates. Pyruvate to lactic acid if oxygen is not present, and pyruvate to acetyl-CoA if oxygen is present. During aerobic conditions. In aerobic conditions, when oxygen is available, pyruvate is transported into the mitochondria, where it undergoes oxidative decarboxylation. This is a complex process that involves the removal of carboxyl group and the addition of a coenzyme A molecule. The result is acetyl-CoA, a 2-carbon molecule that enters the citric acid cycle. But during anaerobic conditions, when oxygen is not available, pyruvate is converted into lactate. This process known as lactate fermentation. So in this reaction, pyruvate is reduced to lactate. Here, NADH is oxidized to regenerate NAD+. If you remember, in the step 6 of glycolysis, while glyceraldehyde 3-phosphate is being converted to 2 molecules of 1,3-bisphosphoglycerate, NAD+ reduced to NADH. In this step, that NADH again oxidized to NAD+. Hence, the balance between NADH and NADH+ is maintained in the cell. This regenerated NAD+ again will be used in glycolysis. Isn't it interesting? Just imagine the level of harmony cells maintain. This reaction is catalyzed by LDH or lactate dehydrogenase enzyme. It is an redox reaction, so enzyme has to be dehydrogenase. Lactate dehydrogenase or LDH exists as a tetrameric enzyme composed of subunits, and there are different isoforms or isozymes of LDH. These isozymes are tissue-specific variants of the enzyme, and their presence in different tissues reflects their role in various physiological processes. LDH isozymes are designated by numbers, and the most common ones are LDH1 through LDH5. LDH1 found predominantly in the heart muscles. LDH2 predominantly found in the spleen and in the red blood cells. LDH3 found in lungs. LDH4 found in kidneys and pancreas. And LDH5 predominantly found in liver and skeletal muscles. When we exercise intensely, we have seen that after some time, muscle activity decreases. This is called muscle fatigue. It is caused due to the fermentation of pyruvate in low oxygen condition. Pyruvate get converted to lactate, which causes muscle fatigue. The Cori cycle, also known as lactic acid cycle, is a metabolic pathway that describes the interconversion of lactate and glucose between the muscle and liver tissues. The Cori cycle is particularly significant during periods of intense physical activity or when the body is in a state of low oxygen availability, that is anaerobic conditions. So, what we are getting from this picture? Muscle cells take up the glucose and use it as a source of energy through the glycolysis. During intense exercise, muscle cells undergo glycolysis to produce ATP for energy. Pyruvate, the end product of glycolysis, is converted to lactate in the absence of sufficient oxygen. Lactate is released into the bloodstream. Then, lactate is transported through the bloodstream to the liver. In the liver, lactate is converted back to pyruvate. Pyruvate is then used in gluconeogenesis, a process where glucose is synthesized. The newly synthesized glucose is released into the bloodstream. Again, that glucose is taken up by muscles. The cycle illustrates the dynamic interplay between different tissues to ensure a steady supply of energy substrates, emphasizing the adaptability of cellular metabolism. So by this, we are in the last few seconds of this video. In the next video, I shall be covering the formation of acetyl-CoA from pyruvate, the other fat. Till then, be happy, stay curious and have fun. Adios.